and take it away.
Sure. Thanks for everybody being here. I'm Xiaoxiao from Columbia University. So it's very nice to, on the light of the week, that we have like another like Nobel laureates winning Nobel Prize because of AI. So today I'll go talk about how AI powered neuroengineering to have like an overview of the current trend and also the future perspective. So before we started, actually, I would like to talk a little bit about my personal journey. So I graduated from Brook College with a dual BS degree in economics and mathematics. Then I graduated from undergrads. I work as a research assistant at Columbia University Medical Center, affiliated with New York State Psychiatric Institute, where I work with many clinicians and also fMRI experts. So I get very interested in and also excited about how the brain is working. So I joined the Columbia University as a graduate school. Then after my internship as a RA, at the biomedical engineering department link lab. So currently I'm also working as a student researcher at Google DeepMind. So I think based on my experience, as you can see that from point A to B is not always a straight line, as you can imagine. So actually it takes many complex paths in order to find what you are really likes and also what makes you passionate about. So I always believe that you are shaped by your experience. So don't be afraid to explore new things and also don't let the fear of making mistakes to hold you back. So finding and also pursuing your passion is never a waste of time. So if we take back to the main topic of today, so let's talk about the science piece. So first, I would like to give an overview of the AI in neuroengineering. So one of the most common application would be the brain-computer interface. So by reading the brain signal from the combined with the machine learning piece in real time, we can actually use this brain signal to perform some tests on the computer. Then a very adjacent but similar field would be neuroprosthetics. So now instead of like using this neural signal to control a computer, we can actually use neural signal to control the robotic limb. Then you can take a look at how neuroimaging would, because in the previous two topics, it's always about like how the neural recording itself. But now if you take a look at the neuroimaging instead, there are many applications of AI on it. So a common application would be how to do the image restriction or image segmentation. So with the help of the AI, the researcher can achieve a faster and even more accurate segmentation to attend to the region that would be relevant to the clinic diagnosis or relevant to the text identification. So those advancements in the AI are going to be super helpful to achieve a faster solution. And then for the neural data interpretation itself, actually with now the term called explainable AI, usually instead of like having a black box to understand how those like machine learning model is working, now everything can work as a white box. So instead of like having those term uninterpretable, those interpretability of AI model itself has improved. And now it's can change this transparency between how clinicians work with scientists to have a better understanding of like how the brain is working in general. And then still very similar, but I would say now the focus would be slightly different. Is now actually we can build predictive model on top of the neural recordings. So for example, a very good example is that people can use neural recording to do disease prediction and also like for some like test prediction. So now with the help of AI, this disease prediction process can significantly be improved. And also it can change some like underlying, like fundamental understanding of the underlying neural mechanism of the brain itself. So lastly, this is a relatively new field, is that instead of like focusing on the instead of like focusing on the prediction itself, now we're focused on the personalization. It's actually relatively new in terms of like how it's being applied in the precision medicine. So for example, if you do have like a lot of like neurostimulation has been happening. So now with the help of AI, we can use the tool to train certain parameters that's going to optimize your performance. So instead of having this unknown feedback loop for the neural stimulation, actually we can build a well-known closed loop feedback based on those like terms that we learn from AI model to achieve a better performance. Basically, it's like pushing forward how we would perform this precision medicine. So first, let's take a closer look at the BCI. So based on the distance from the recording system to the actual neuron firing, we have different kinds of input for the BCI. On the scalp level, we have EEG recording, which will record brain activity non-invasively. Additionally, if we have some go deeper, we have the recording on the brain surface interface, so that will be named as ECOG. And then we have something even more invasive approach involving microelectrode. So this method is going to allow us to record brain activity at a very fine scale, capturing local field potentials and neuron spikes, providing a high detailed insight into the neural activities. But nonetheless, all of them combine with AI model to control the for the task on the computer. Then, if we take a look at the neural imaging instead, unlike the EEG and ECOG, which have a very good temporal resolution, the neural image usually have a better spatial resolution to indicate the brain activity at specific brain location. So a common example here of the neural image would definitely be T1, T2 fMRI, and also PET scan. So in most of the time, people also classify the functional MRI as a neural imaging tool as well, although its spatial, if you compare to other recordings here, so its spatial preciseness is not as good as the others, but actually it still intends to specific regions of the brain on the voxel level for us to understand the brain better. So if we take a look at the application side of the neural imaging, first, we can think of the image enhancement. So when a researcher obtains a brain image with a very poor resolution, we can use a generative model to reconstruct a higher quality of the image and potentially denoise the image to have a better brain signal to noise ratio that will benefit the downstream analysis. Basically, you can take it as a denoising process. And another example would be the cross-referencing between modalities. So when you combine, when you can obtain one type of image, such as MRI, you can use AI tools to generate the participant's PET scan instead at a very low cost, because now you don't need to actually perform this PET scan. You just need to use AI model to predict how the PET scan look like. And also it would benefit a lot to many facilities who actually don't have all type of modality available. Now this transferability between one modality and another modality would definitely benefit the patient itself and also benefit the diagnosis process from the clinic side. And then if you talk about the prediction side, so scientists also trying to classify this detection of the brain structure abnormality as a predictive task, because now for a given structural image, we can use the AI model to segment the brain location and predict the existence of a tumor or some like other brain abnormality in general. So now you can use this brain image for the disease diagnosis directly instead of like just looking at the entire neural recording for diagnosis. And then with the help of the AI, we can also learn these features with a lot of prior. So now instead of like just like looking at this brain image, reading by the clinician, and then do the diagnosis process. Actually, if we do have like enough prior of this neural disease recording, actually for a given image, we can easily predict if this participant have certain disease or even the severity level of the disease, which is like the disease progression. So this is not only limited to some structural changes in the brain. So even with a functional MRI, we can also perform this accurate diagnosis process. So now if we align what we have discussed previously for all the topics that involve the AI in neuroengineering, I would like to introduce 3 projects at our lab based on their different inputs and also different methodology. So first, I will talk about the project using the gaze information to inform a visual transformer to implement human guided AI model in order to achieve a better motor performance. Then we will take a look at the study that actually focuses on the fMRI. For that, we will leverage the concept from matrix learning, which is actually a similar idea as contrastive learning, to try to learn those representation which minimize the inter-class differences and also at the same time maximize the between classes differences. Then in that scenario, we can have like better performance to achieve our prediction, because now the distance between the representation difference between similar classes is actually smaller, and then the representation between different classes is actually bigger. So that's going to benefit a lot for the classification process. And lastly, we will still talk about the traditional inputs for the neuroengineering, which is the EEG. And instead of like using EEG task to do the task prediction, for example, if you have like the auditory oddball, you use EEG to predict it's a novel sound or standard sound. Actually, instead of like doing that, here we focus on to apply this like AI algorithm to predict the underlying context of the participant itself. So now we use this brain activation during the test to distinguish a patient from a healthy subject. In this case, we're going to focus on Parkinson's disease patients. So before I started about the vision transformer, so I would like to give a brief overview of what a vision transformer is. So if you guys do familiar with the transformer model, you can see like the transformer takes the inputs as sequentially and then goes through the multi-heads attention process and then go to the to construct the encoder and then use it to combine with the decoder side to have a performance. So for here, the vision transformer would work slightly different, because now instead of having a sequential input, you can see that the image itself is a 2D space. So what people have been leveraging on that is now they flattened this representation in 2D space into a 1D space presentation. So now you can see if we do have an image like this, we can separate it as a 3x3 grid and then flatten it out with those position of those grids. So you can see that now, instead of like only having those tokenized flattened patches, we also have those position embeddings. So then it's going to work as an input for the actual transformer process. And for the vision transformer itself, so the transformer encoder side is actually the same as a traditional transcoder. So as you can see that after the embedding and also the flattering, combining those like transferring from 2 dimension to 1 dimension, it goes through the same process to go to the norm and then go to the multi-head attention to compose this decoder side. So what's its achieving is that now for a given input, so for example, if I give you image as a dog, the one example on the very top, so the attention map gonna give you the information of, oh, actually for the entire image, I should be attend to those locations that's actually relevant to the dog. So now you can see the attention map is actually lighting up those main object, which is main target for a given image. So on the very bottom, as you can see, now if an image gave you a snake and also a leaf on the bottom, it's very hard to see if I need to more attend to the snake or I need to more attend to the leaf instead. So basically you can see now this attention map is actually giving an equal balance between the two objects. So how are we going to use that? how we're going to use that in our experiment is that, so we collect data in a VR experiment. So now participant is driving in the VR experiment. So when you're driving, your eye is moving to attend to specific location that would be relevant to your decision making, right? So then in that scenario, we can, instead of like using this attention mask that generate directly from this image, we can also use a pupil-guided attention mask. So now those gaze pattern exists in participant is actually indicates of where I am attending to when I make my motor decision makings. So that's kind of like the main idea drive our experiment design. So we're also trying to simulate these results on top of actual environment. So this is an open access data set. So basically now people also driving the highway. Then we have the gaze information being recorded, and then that's going to also used as an input to inform how my vision transformer should be trained. So here is an example. You can see that now for the peripheral masking, so basically we take the gaze patterns and then also adding some like residual possible visual locations around that gaze patterns. So that composed as a peripheral masking. So now we use two types of training loss. So one is joint scene fixation and another is fixation attention intersection. So the main takeaway of this slide is that you can see instead of like now this attention maps and also generated by the vision transformer is overlapped with the fixation map, which is generated by the gaze data. So now you can see there is a significant overlay between one another. And that is the region that we really need to pay attention to, because that is kind of like cross-validated from both vision transformer attention and also human attention. So here is an example of how those fixation distribution would be informed and integrated from a vision transformer. So you can see on the left is a example of a low uncertainty image. So you can see all those objects in front of the driving wheel is very clear to the participant. And now you can see here, the participant look at specific location. And what we're doing is now we overlay this attention mask with those training process of the vision transformer. If you look at the end, you can see now at the time when the visual clarity is very high, basically you can see what is happening during this experiment. Your vision transformer is actually in the end overlaps a lot with your fixation map, which is your gaze information that is provided by the participant itself. So you can see that overlay is naturally occurs even at the training process when the visual stimuli is clear. And another example is that if we do have like a high uncertainty where the froggy level of how my driving scenario is different, so then at the time, my visual clarity is less. So in that scenario, my gaze is actually not helping me a lot because I already don't know what I need to do to go through that scenario. So you can see in that scenario, the overlaps between the gaze and also the vision transformer output would be slightly different. So you can see this overlap is less, but also it's still trying to attend to that relevant location, which will provide you the most information to make the decision-making of your downstream task. Here is the output of that. So if we do integrated those human-guided vision transformer, we can find that it actually have the best performance compared to other types of model. So we also generated a peripheral window, which is the one I introduced briefly before. It's just like adding some like additional regions on top of the human gaze location, and also compared to the random window. So it definitely means that the human guided gaze pattern that's used in the transformer is giving you another level of information that's going to be very helpful for your decision making. So here we use this information to predict the motion action for those participants. So basically now in around 70% or even 75% 90% of the time, we can make a good prediction of how the next motion would be for this participant. Then I would like to switch gear from the transformer to transformer with gaze information to matrix learning with fMRI instead. So for the fMRI, as you can see, it have like a lot of like precise recording of specific location. So there is a significant overlap between the variability of the both activation that you observe in the healthy subject compared to the disease population, especially for the functional MRI, because it's not all the functionality of the brain would be different when you has been diagnosed for certain disease, right? So basically what we want to achieve is that there might be a dimension reduction methods, for example, like Princeton component analysis, would be able to capture those shared variance, but also at the time when you're trying to decrease this generalization, you also can like decrease this, trying to increase this generalization, you also try to decrease this discriminability between healthy variation compared to the disease variation. So ideally, as you can see, we want to generate a latent space that can naturally separate those healthy subjects compared to the disease subject. So as I described briefly earlier, is that the entire motivation of the matrix learning is that we want to have a high discriminability, which means we have enough distance in the representation space between the healthy subject compared to the disease subject. And also we still want to keep those generalizability because we still want to have this ability to interpret what is happening in those brain, right? So this variance in data would capture by the embeddings that we obtain from the latent space. What we are proposing to do, as you can see here, we show you the overview of the information mixed learning, is that now with a specific fMRI volume, you can separate it as a 3 presentation. So one is called positive anchor and another is called negative anchor. And then you also have like anchor have like no labels, which indicate nothing, like basically the benchmark anchor you can interpret as that. And then all of them goes to the fMRI encoder, which is a f theta dot function here, and then generated to the latent space. So what is happening in the latent space, as I described earlier, is that now in the latent space, we want to maximize this distance between positive anchor and negative anchor. And also at the same time, for the negative anchor itself, we want to decrease this distance between negative anchor and also maximize the distance within the positive anchor. So in that scenario, as you can see in this figure here, it's naturally falling into 2 representations. So one is belongs to the positive anchor representation space, another is belongs to the negative anchor representation space. And then if you go through a decoder, you can also reconstruct those anchors in this space. So what does this mean? If you look at this latent space that we know, so we do this algorithm application on the people on the ABD 150 dataset. So basically that is a dataset with healthy subject and also people with autism. You can see that the representation space that we know, which is also can be called as latent space, is that now the latent space naturally falling into 2 categories. So one is represented by the red color, which is a people with autism, and another is the healthy subject location where the green dots. So you can see it's naturally falling into 2 categories. The overlap between one and another would be minimized. So if you look at another representation that we generated from, for example, the principal component here, the entire latent space is actually overlap with each other a lot. So you cannot have like a very good separation on top of this latent space because it's essentially not falling into 2 categories, basically overlap with one another. A lot. So based on the 5-fold cross-validation result that we found, the machine learning algorithm we are trying to imply here have the best performance to separate the autism disease from the healthy subject in this scenario. But also, as I briefly mentioned earlier, is that we still want to keep this interpretability exist in the FMI, because if I'm a clinician, if you diagnose us for a certain disease, there would be a reason underlying that, right? So there is no way I would just like use my AI predict model says, oh, I think you have autism. So basically, now you still want to have this interpretability exist, especially for the clinician for better diagnosis and also for the neuroscientists to understand what is actually goes on in the brain. So basically, you can see here that with those latent space, we can still reconstruct what is happening in the original space, which is the fMRI space. So you can see it still have like some joint features that share between the diagnosis with autism. You can see those post-occipital region that's in the brain still have like very strong activation. So that's actually linked to the clinic symptom in autism disease patients. So lastly, we gonna talk a little bit about the disease detection. So in the previous, we use an open data set. For here, we use the open data set as well. But now instead of like separates people from autism to people just like being a healthy subject with FMI, now we use something, we use a test EEG to separate people from people with Parkinson's disease from people just like being a normal control here. So I would like to give you a small introduction of what is a Simon conflict test. So basically, a Simon conflict test is a cognitive test that has been run for many years. And there are two conditions was defined in Simon conflict test. One is called congruent trial, another is called incongruent trial. As you can see here, a participant would be provided with a stimulus on top of the screen. So if I indicated, for example, if I indicated a red button or the yellow button on the right side of screen, I need to click the right side button to show my spatial consistency. So that's kind of like a congruent trial is defined. And then if I do have a blue button show up on screen, so based on the instruction, I should click the red right button because it should be indicated the right side of the screen. But instead, I need to still click the left button. So that's kind of like naturally form a spatial inconsistency. So basically, a hypothesis why we use this test is that the hypothesis is the people with Parkinson's disease would have like a longer time to react to those response after the stimulus has been showed up. But actually, when we run this comparison in terms of reaction time, we didn't find a significant differences between people who have Parkinson's disease compared to people who are just like a healthy subject. You can see the ROC curve is actually around 50%, no significant differences. And then we are thinking of like what we can do. So if we don't have like a behavior level differences, so maybe some like brain level differences that we can use to help with the diagnosis process of the Parkinson's disease, right? So what we are doing right now is here. So we extract those spatial temporal features out of the 64-channel EEG recording. So we reject one channel. So we actually didn't perform this text ourselves. We use an open access dataset. So feel free to try it out after the code demo session to kind of like check it out how the data set look like and how you can rerun some of the code on top of the data set. But a brief idea is that here we take the temporal and spatial features out of the EEG recordings. So after the onset of the stimulus has been shown, we take the 1 minute right after the onset and then use that as the input, which is a temporal input for the representation of brain activity changes. And another representation is spatial feature input. So now instead of like taking those temporal feature, which is a time series itself, actually we calculate this correlation between one electrode to another electrode So instead of like focus on the brain activation of certain region, now I care more about one region's relationship in terms of another region. So now there are different types of like index that you can calculate on top of that. But basically here is an example of the Pearson correlation. So you can see that now we can generate a symmetric matrix that represents the relative relation between one electrode to another electrode. So that can actually collapse everything in time into a spatial representation instead. So if we do take those temporal and then spatial representation as input for our model, based on how the feature itself is looking like, we can like choose different architecture to combine with it. So first, we try the random forest, which is basically you can try it on all types of different input features. And then we have long short-term memory. So long short-term memory is specifically probably only apply for time series data. So that's where we're going to try it out on the temporal feature that we selected from the EEG recording. And then we have CNN. So CNN is the convolutional neural network. So basically it has been used to allow like image recognition. So actually that's kind of like align with our spatial feature that we selected computed from those EEG. So basically there are 3 models that we test, and also we have like different layer combination. So you can see the parameter size is slightly different. So here is the outcomes. So in order to have like the best performance, we definitely do a little bit pre-processing to the data itself. So now instead of like focus on the entire raw EEG data, we filtered it to only obtain the data band, which is 124 Hertz. And then based on the output, you can see that no matter for the congruent trial and incongruent trial, actually this representation from spatial combined with random forest classifier is actually giving you the best performance. So in the most of the time, probably 95, 98% of the time, you can separate one trial performed from Parkinson's disease patients to one trial performed from the healthy control patients, healthy control subjects. And we also tested on another band. So usually on top of delta band, people also test because it's involved the cognitive part. So people also test on the theta band, that is 4 to 7 Hertz. So you can see that even for this, we still have the best performance that is represented by the spatial features. So you can see that now, although the spatial and temporal information carry some like same level of information to indicate whether this participant is a Parkinson's disease patient or a healthy control, actually we still have some advancement in the spatial representation itself to explain how those like relationship between one electrode to another electrode, which can be understood as a functional connectivity, to inform whether this participant have different brain activation pattern compared to the subject who have disease. So here is a brief summary of how the subject level output. So as you can see, the previous result is about the trial level. Basically, based on a given trial, we want to distinguish whether it's performed by a Parkinson's disease patient or healthy control. For here, we actually run everything on a subject level. So now we have 75 subjects has been tested on this algorithm. So you can see that for the reaction time, which is indicated by the gray bar here, there is no significant differences between the control and the patients. So as you can see, the probability here indicate how likely I am gonna classify this participant to be a Parkinson's disease patient, right? So when it's closer to 1, it's definitely I am more certain about classifying this participant to be a Parkinson's disease patient. And if I'm closer to this probability is closer to 0, I'm more confident to classify it into the control group. But for here, you can see the RT is not working as our previous ROC curve show. But for here, something interesting happened. So although in the previous one, we have slightly better performance in the spatial representation space, But now you can see that on the individual level, this differences is even bigger because for the spatial representation, which is indicated by the yellow bar here, is actually more closer to 1. So for a given participant, I'm more certain about my decision saying, oh, this participant should be a Parkinson's disease patient, compared to the temporal representation result is only achieving maybe 70%, 80% about the certainty of whether this participant should belong to the Parkinson's disease patients or not. So if you're looking at the control side, which is the first 25 subjects here, so you can see that even for the control, the spatial representation is more certain about, oh, this participant should be actually be a healthy subject compared to the temporal representation, where still at times it's still close to 0.5, which is indicating, oh, I might make a wrong decision. But overall, it still naturally like separate the patients from the healthy control. That's why a lot of study is not like discussing how the temporal feature and spatial feature can have like benefits from one another. But for here, at least like we can see that although individual level, we can have an accurate detection of the Parkinson's disease, no matter I'm using temporal feature or the spatial feature. But at least what here is shown is that with the spatial feature representation, I have more certainty of like my decision out of this entire model. So lastly, I would to talk a little bit about the future direction. So as you can see, like the large language model based on also recent like hot topic about Nobel Prize. So basically, definitely open large language model is next thing to go in those like AI in neuroengineering field. So the Google had the GMA model, which is open source, and also the Meta have the Llama model. So definitely a lot of like researchers are currently trying to integrate that pieces into their own project to have a better performance. Another thing is to use a physics-informed AI. So as you can imagine, a lot of like neural activation precisely is bounded by some like physics terms, right? So basically, if I do write down the Hodgkin-Hoskin model, I can actually use those PDEs to calculate how my neuron firing is performed. So those activation in the brain is naturally bounded by those physics models. So now instead of like just using AI itself, we should use physics-informed AI. So basically, there are two examples that are listed here. One is called PIMS and another is called InvestNet. So basically, two of them is using those those physics features of those models and then trying to have a better performance and also a more natural performance, because now all those like learning process is bounded by the physics term physics model. And lastly, so this is another big hit recently about the CAM. So basically, in the previous multi-layer perceptron, the MLP, you updated the weights on the neural itself. So basically, now with the CAM, instead of like update the weights on the neuron itself, you actually update the weights on the connection between one neuron to another neuron. So basically, it's have kind of like form a new way to updates to have update out my weights for my AI models. So that's kind of like very interesting because actually they based on the paper they state they have better performance, and it's actually links back to the physics informed AI a little bit, because now all the thing that you learn is more certain. So you basically can achieve a faster convergence out of that. So here is the references for the slides. And now, before I go to the demo, is there any questions?
Thank you so much, Xiaoxiao. Wonderful presentation. And yeah, we're definitely, for anybody that's in the call, if you have any questions for Xiaoxiao, please, you can unmute or you can put your question in the chat.
Yeah, no worries. I think I can share the code first.
Okay.
So I'm sending the code link to the chat so people can access it. And now I'll share the screen for the code.
And yeah, it looks, I was able to access it, so hopefully participants can as well.
Okay, great. Can you see the code?
Yes.
Oh, great. So basically the code itself, actually we take the example of the last project. And if you go back to the, you can directly, so as long as you have like a Google account, you can directly run it from Google Colab. And let me see. And basically this code is actually taking a partial code from actual paper and also partial data from actual paper. So it might give you like completely different results. So don't try to kind of like implement the paper outcome with this existing code. But basically in this code itself, we'll go through certain parts. Let's see. So one is an initial setup. And then it's gonna tell you a little bit about how to load the library, how to define functions. And for here, we're gonna briefly talk about the long short-term memory and also the convolutional neural network. Here we use in the paper. And then we have the data loading part. So definitely download the data here if you haven't done it, and then put it in the Google Drive so we can directly mount the drive and then do all the loading and then all the computation later. So then we're gonna, before we actually start training, we actually need to define some like model inputs. So basically you need to have the data set merged, which is gonna generate our input for the model itself. And also you need to define your input layer, kind of like make it align with the model itself. And then you do the training testing split to split your data set into two sets. So that's gonna benefit you a lot on your training process. And then we're gonna start the training. We're gonna talk a little bit about the enfolds, cross-validation, and then how to update. So usually at the time you need to have like some like hyperparameter, or sometimes not even hyperparameter, just like to want to find the best validation model used as your final model for your test data set. So we're gonna teach you a little bit about how to update that. And then we're gonna show you the testing accuracy, how to achieve the, how to get the testing set accuracy. And lastly, we're gonna do some little visualization about like how you should plot the training result and also how you can print all the model parameter as your summary for your model. So first, we're gonna do the import. So all the imports is kind of like import all the necessary library that you will need it for your calculation. So we're gonna let it run in the background and move a little bit to the lower part. So now, as I mentioned briefly in the slides, is that we're gonna use long short-term memory and also convolutional neural network for our training. So the long short-term memory is a type of recurrent neural network. So as we discussed, it's kind of like takes the time series as an input, unlike the convolutional neural network usually take a 2D space as an input. So based on the time limit, we will not go through all the details about the long short-term memory itself here, but basically the long short-term memory you can thinking of now, you have a lot of dependency between the current moment to your previous moment and also your future moment. So basically the structure, as you can see, is indicated by the t-1, t, and then t+1. So basically that indicates your time consistency in your model. And also, a important part is now for the long short-term memory, you have different gates. So one is for the t-1, so that's kind of like what is happening in the past. Combined with what is happening right now, xt, it's kind of informed or forget gate. And then you take everything to the input gate, and then after the calculation process, you move to the output gate, and then the output gate gonna combine with the future information and then to inform your future state. So basically, feel free to go ahead and read more details here. But basically, I think as long as you understand how each gate is working, you definitely understand like how long short-term memory model is working itself. And for here, then we need to define how to build a long short-term memory model. So basically, as you can see here, we build this long short-term memory with those parameters that we defined here, and then adding some dropout. So the dropout here is usually added for regularization. And then we have another layer of long short-term memory, and then another dropout. Then everything is connected to the dense layer. So dense layer is actually working the same. So we're gonna talk a little bit later in the later slides, but density layer is just like a deep neural network that can like have all the different layers combined and then do the calculation layer by layer. So nothing fancy there, but you can see, so here is an example in the convolutional neural network. So now for the convolutional network, I think most people are more familiar with CNN compared to the long short-term memory because there are a lot more people work on the image side. So basically you take an image as an input and then go through those convolutional process and then generate all those presentation in between. So in the end, you flatten it together to be a one-dimensional representation. So you can see now you flatten everything after the convolution process. Now you flatten everything, and then it goes to the dense layer again. So this is the dense layer that we use the same in the long short-term memory at the end. And then you have the output layer, which is like indicate what your output would be. So feel free to click the link and then read more details about that. And then here for the function, we define how to build a CNN model. So we have all the parameters defined here. So that's basically decided the size of the CNN. And then you flatten everything as you see here as an example. Then you go to the fully connecting layer, which is a dense layer, and then go to the batch normalization and then activation ReLU. And lastly, you have this output layer, which is your classification layer here. So we have another function defined here to create labels. So for the data set that I provided to you, there is no label attached. So we just like define a function here to create a label for those data set that you load. And I think that's going to be the most important step that I want everybody to get on board is that now you actually need to launch a drive. So basically it gives you the access to your Google Drive. So then you will be able to load the data set that you have received from at your Google Drive. So it might take a few more minutes, but then what we can see here is that we define the class condition. So class condition, as I mentioned, for this experiment, we have congruent trial and also incongruent trials. So we only provided incongruent trial here for the comparison purpose. And then for the band, we use actually the theta band results for the data set, and also the amplitude indicate we are using the temporal feature because we're gonna run long short-term memory model first. And then here define some like parameter that's relevant to the EEG dataset itself. So we have the sample rate and also we have the time steps. As I mentioned, we only take the 1 second right after the stimulus onset as our input for the model. And then the channel number, the recorded is 64, but we rejected one channel because of its poor quality. So we end up with 63 channels. And then naturally the features of those channels would be 63 in the scenario. And because of the time limitation, so I changed. So usually people run like 10 folds or even more folds, but now I changed it to 5 to have like more time, less time for the train, more time to go through the code. And then the number of classes, you can see it's a binary classifier. So basically the number of classes would be just 1 here because you are just like doing a 0-1 classification. And then here define the epoch size and also the batch normalization size, and then the patient number is actually defined for the early stopping. So now if you have like a longer patient number, you definitely will go through more round of validation before you reach the decision says, oh, I need to do an early stop here. But that's some like details you can definitely check out later. So what would be most important is that now we need to load this. So if you do want to save the drive, save the data to a different drive location, so you might need to change the path, but you can just like save it in your drive directly. So don't put like names don't put it underneath another folder names on top of that. So then you can directly be able to load this entire folder, and you will be able to load. So the C1 here is actually the trials from the healthy controls, and then the C2 here is a trial from the Parkinson's disease patients. And I give you 500 trial for each condition. So now you would end up with 1,000 trial for the model input. So as I mentioned before, we need to kind of like, because you cannot train, so you need to merge everything together, right? Since you now have like two conditions. So now instead of like having it separately, you need to merge it all together to work as an input for your model. And then you create the label, corresponding label on top of that. So now you are labeling the healthy control as 0, and then the Parkinson's disease patients as 1. And also sometimes you need to adjust the classification weights. But for here, it's not a worry because now we have equal number of inputs for each condition, but sometimes you might have like more trials from the patients and then have less trial from the controls. So basically this is a function that I put it here for you to balance the weights between your representation from each class that you obtain. And then you do the train and test split. So now you use a train and test split with 80% versus 20%. So for here, the test size decided how the distribution you want it to be for your testing and training data set separation. And then let's run the model. So I won't click the button here because it takes some time to run. But basically, if you click the button, it's going to go through the K-folds validation process and then keep updating this model until it's have like the best weights on the validation process. And you can see here is the code to update this model to always keep the one with the best validation results. So up to here. And then they're gonna print it the results whenever they update this result. And also they're gonna validate this result on the testing set that you previously separated. So you can see I already run this block beforehand. So you can see now the average, the kind of like the training accuracy would be 88%, and then the validation accuracy would be 85% at the fold 1. And the accuracy of the testing data set would be 83.5%. So that's kind of like a very good performance in general, since you only have like 1,000 trial here. And then, as you can see later on, the first folds that we run already give you the best performance. So basically, we didn't update this model because no validation accuracy is higher than the first folds. Then we still like at the end, when we re-verify it on the testing set, we still obtain the same accuracy as what we are already shown here. So lastly is the results validation. So usually this step is done before you actually run the model, to at least like to know how your model architecture is. But basically now we can print it out the model layer itself and also combine with like the number of parameters that we have. So basically now you can see the size of this model that we train is actually not very big. It's very small compared to especially those like large language model that we have. So you can see it's only have 9,000 parameter tuner in that scenario. And also there are some like even non-trainable parameters. So like the overall, like that's why like in general, that's already computed relatively fast compared to many big model. So then you can plot the training and validation accuracy here. So you can see that usually in most scenario, the training accuracy is always higher than the validation accuracy, since like you tend to fit the model until it's kind of like converges at a stable level for your training accuracy. And but also like at the time, if you do if you do have like very high training accuracy, for example, the FODS1 here, you can actually this should be FODS 2, but you can see that when I have a very high training accuracy, I actually don't have like very high validation accuracy, because at this scenario, I might have like a problem called overfitting. So it means I overfitted my parameter to make it predictable in my training set. But now if I move to my validation set, those parameter is definitely overfitted because I don't have like as good performance as the training data set is. And then later part is still the same process, but now instead of like loading the temporal features, now we actually loaded the spatial features. So that would be 63 times 63 for each trial. So here we take another example. We didn't use the incongruent trial. We use a congruent trial as an example, and then we take the delta band for my as a target frequency band that we want to work with. And then for here, we use a Pearson correlation data. So actually there are other spatial features that we selected to do the representation in the paper. So you can read the paper for more information about how to calculate the spatial representation out of the EEG data. But basically those parts are the same, just like define some like EEG related parameter and also model related parameter, and then you load the data. So now I still give you 500 trials from each representation, but you can see here, if you merge it together, the input size should be slightly different. Previously, we have 1,563, but now instead of like having 500 time point, now we have 63 electrode, because now we change everything to a spatial representation. And then you do the train test split, and now you still train the model. So basically everything is the same, except now you call the function to build a CNN model instead. And then you keep updating the model to make sure it is the best. So as you can see here, this is definitely a very good example of overfitting. So you can see now my training accuracy is very high, but my validation accuracy is very low. So it's definitely not even as good as random guess. I would say like it's probably equivalent performance as random guess. But basically, this is a very good example of like overfit. So actually, in contrary to what we obtained from the paper, this little demo actually give you a contradictory finding, because now it's saying, oh, actually, if I just like using the spatial representation, it's actually perform worse because I tend to overfit my model from my training set, and then you don't have like a very good performance on my testing set anymore. But as you can imagine, it only give you 1,000 representation. So that's a very limited number of trials. So actually in the actual paper, we will use like up to almost like 30,000 representation, like trials representation from the entire data set. And then lastly, we still do this result visualization. So you can see now the CNN model definitely have way more parameter compared to the long short-term memory in terms of size, but also like based on how the CNN, so this might be like not so relevant, but based on how the CNN and the long short-term memory is constructed, actually the computation process is fast, even though there are more parameter to fit, but the computation process is actually faster for the CNN model in order to finish the entire training. And here is a visualization of the plot. So you can see this is definitely a good example. As I mentioned, over and over again, it's about like the overfitting. So you can see the training accuracy is very high, but the validation accuracy is very low. So that's pretty much about the code demo, but feel free to run through the code and let me know if you have any questions.
Hi, Xiaoxiao. Thank you for the presentation. It was like very detailed. And then I have a question just for the validation accuracy. So what's like a good validation accuracy for the task?
You mean what kind of like accuracy? You mean?
Yeah, like validation accuracy. What's like
validation accuracy. I would say like usually if you have like training accuracy at like 95%, you usually expect your validation accuracy achieve like 90% as well.
Okay.
Yeah, so usually it shouldn't be have like a big misalignment, because in that scenario you definitely overfit, but also like it depends on like how much sample you have. So if you do have like a small sample size, so it's usually happen you don't have like that many high validation accuracy. It can goes up to like 10%, even 20% differences, but that might not be an overfitting problem. Just like for the architecture design itself, it's might only because you don't have like that enough sample to train the entire representation. So it might not be a problem in your model itself. It's only because you don't have enough sample to train your model, basically.
Okay, thank you. And also, could you like explain the Simon conflict task again? Because I was like doing experiment, I missed some parts of it.
Oh yeah, sure. I can quickly show here. So for the Simon conflict test, is that now you have some stimulus shown on the screen, right? So basically, for a Simon conflict task is about the spatial consistency and spatial inconsistency. A spatial consistency is that if it's stimulus shown on the left, and if the stimulus is indicated to responded with left button, so then you respond with a left button, that would be a spatial congruent trial. It means I saw something on my left and then click the left button to indicate a correct response, right? And then for a spatial inconsistency, that would be an incongruent trial. Basically now, when I see something as blue here, I should, based on the instruction, I should click my right button. But now, since I'm in an incongruent trial, I should click my left button instead. So now it's a spatial inconsistent response, right? Because I should be expecting seeing something on my right, and I'm indicated to respond with my right hand, but also at this time, I'm in an incongruent trial, so instead of responding with my right hand, my correct response should be respond with the left hand. So that's kind of like how the spatial inconsistency is formed.
Thank you so much. And then like the coding is like very detailed. Thank you so much.
No problem, no problem.
I have one related question about this. So did you say that the hypothesis is that for the Parkinson patients, there should be a higher or larger response time? And so,
yes.
So would you be able to, like, with your results that you show later in the talk, that there is more evidence for the hypothesis to be true? Like, it is still a hypothesis, right? That's why
I think there are studies that has been shown the Parkinson's disease definitely have higher reaction time, but I think that's kind of like varied by how much. I don't remember the exact number from that paper, but there are papers prove that the Parkinson's disease should have a higher response time compared to healthy subjects, but also it might because they only run like small number of subjects. So for here, I think the conclusion might be more validated because it have in our calculation we have 75 subjects, so that's kind of like a lot. So basically, in our scenario, across all those trials that be performed by those subjects, there is no significant differences between the reaction time between the healthy control and then Parkinson's disease patients.
All right. Thank you so much. It was really
no
nice
problem.
talk.
I think probably it's about like the group level differences. Like, if you run it on different type of population, it might be different. So it's kind of like also biased a little bit based on the population, like the clinic cohort that you are working with.
I see. I see.
Yeah.
Thank you so much.
No problem.
Thank you so much, Xiaoxiao. Wonderful job. I'm going to stop the